to go through a little bit of the presentation, which will be held at the AIRS conference in Barcelona. And my topic will be to discuss the role of the nasal microbiome in health and in diseases. So first of all, it's of course necessary to understand the differences between whole shotgun metagenomics and 16S PCR as a non-specific way to analyze the human microbiome. Afterwards, we will go through some differences seen between nasal swabbing, so different kits actually used. Then we will go through the different ways to do so, so brushes versus washings, and further head towards contamination issues, which has been seen a lot in performing more sophisticated analysis of this low biomass microbiome. Afterwards, we will head towards the composition of the human, let's say, healthy microbiome, and see that diversity is actually much more present in the healthy microbiome. Afterwards, we will briefly touch some diseases. So first of all, chronic rhinosinusitis, and then head further to atopic asthma. Afterwards, we will start to discuss one of the diseases I'm very interested in, which is called granulomatosis with polyangiitis. It's a kind of ANCA-associated vasculitis where the body is actually producing antibodies, and antibodies are directed against vessels. It has been seen that in patients with GPA, actually nasal contamination and nasal carriage of Staphylococcus aureus plays an important role. And so our issue was actually to perform a pilot study actually analyzing the human microbiome in this kind of disease. And we found that even though Staphylococcus aureus was more present in nasal culture and in disease activity, this actually was reduced during disease remission. And among the human microbiome analysis, we found that there are novel strains which hasn't been published so far in humans. So we identified Staphylococcus pseudointermedius, which is a novel strain, by culture and also by more sophisticated genomic analysis, and published this last year. So we will head further to novel sequencing measures in the nasal microbiome, and then we will briefly discuss some of the future directions and how we can change the nasal microbiome, so either by local treatment with steroids or local antibiotic ointment, and then further discuss one study which analyzed the impact of systemic antibiotic treatment on the nasal microbiome. And so we then will conclude the presentation with a brief outlook what will happen in the future. And with this, I would like to conclude my short introductory video, and we will see each other in Barcelona.